What about AIDS treatments? And this has come a long way, actually probably over your lifetimes now that I think about it. When this first got heard about in the mid-'80s, I was in college, and there was a buddy of mine in lab who was thinking, this could be the science fiction disease that wipes out all of mankind. Because when it was first known in the early '80s, there was no treatment, and as people were extremely infectious. It's not amazingly contagious, but it was uniformly fatal, as far as anybody knew. And so it looked pretty bad in those days. But because of understanding about how viruses work, we were able to mount relatively effective treatment plans. And in fact, they're working towards the vaccine. So it does not look like the science fiction disease that will wipe out mankind, only because we were sort of on the ball with it. So these are all new, sort of since you're more or less in most of your lifetime. So when you were little kids, it was much more risky than it is today. It's still a dangerous disease, but there's a lot more options than there were back in the early '80s. So now one of the first set of treatments that came out were antibiotics for the opportunistic infections. So the first things, they just dragged them right off the shelf. And one of them was something called rifampin. What that is this inhibits RNA polymerase of prokaryotes, that is bacteria, but it has no effect on eukaryotic, that is human, RNA pol. So, because eukaryotic and prokaryotic RNA polymerase are very different. And so as a result, it kills the bacteria only. So the way this works is that there's been at least 1 billion, 1.5 billion years of evolution between bacteria and us. And so our RNA polymerase is really quite different than theirs. Our promoters in bacteria would not work in humans and vice versa. They're really quite different proteins. Just a second. Different enough that you can make a drug that binds to prokaryotic RNA polymerase and shuts it down. So then those bacteria can't make any RNA. They can't make any protein. They're dead. All right? Doesn't stop AIDS and the virus replication. Doesn't stop the underlying course of the disease. But at least it can treat those infections so that the person survives that infection, and was at least a way to tide people over until something better came along. Did you have a question? Yeah. So the question was, HIV can be dormant, and you pass it on to your child. I want to be very careful about that. You don't pass it on through eggs and sperm. It's through bodily fluids. They don't inherit it from you. They get it from blood or whatever that happened during birth. Would it be dormant in the kid? It depends on I don't know the factors that influence whether something is dormant or active or not. It depends on I actually don't know what controls that. All right, yeah. There's a question, can you test a kid that young to see if they're HIV positive? To be honest, I don't know. I would presume the answer is yes, but I don't know. Yeah. So this is not a great win either, as you were saying, because eventually the bacteria will become resistant to it. Although if you hit them hard enough, oftentimes they die, and that's okay. Antibiotics usually work. All right. So that was one. The problem with this is, but there's no effect on HIV because it's not a bacterium. So the next set of drugs were developed based on actually an understanding of the virus. So number 2, targeted to parts of HIV that are not present or needed in normal human cells. So as a result, therefore, you kill the virus, not the host. And this is another interesting story because retroviruses were for a long time a curiosity. Back in the 70s, there was a thought that retroviruses caused cancer in humans. Turns out they don't, but there was a ton of research done on retroviruses in the 70s in the hope that it would be a lead on cancer. And it turns out it wasn't. It was totally a dead end for treating cancer. And so all that stuff got kind of put on the shelf, and they thought, oh, well, it was interesting, but we didn't help with cancer. But then when it was discovered that HIV was a retrovirus, they said, excellent, we have all this stuff on the shelf, all these articles and information we can pull off the shelf because we know how these things work now. And so we were way ahead on AIDS, even though that research hadn't helped with what it was sort of intended for, which was cancer. And the first set of drugs that came out targeted reverse transcriptase. That makes the DNA copy of viral RNA. And this is related to DNA polymerase. It's something similar to that, but it's not present in normal human cells. Right? And so if you look at the next page of today's information packed handout, there's a couple of chemical structures of drugs that were used for this. So let me show you. If you, it should be there. Yes. All right. So let's see pages. So there's a couple of them that I want to focus on. So people have probably heard of AZT, right? That's a sort of common, that's 3 letters that have made it into people's consciousness. Was one of the very first. Another one, DDC, most people probably haven't heard of unless you know somebody who is infected with HIV. That's another drug that's used. Right? The drug off the shelf is this thing without the phosphates. If you look at these and then go back and look at some earlier handouts, you'll see that these things turn out to look a lot like ATP, and that's not a coincidence. ATP can be used as a building block for RNA, and these are actually building blocks for DNA. The thing that's really important and interesting about these guys, the thing that they all have in common, is that if you look at the normal, they have a 3' OH, so CTP and TTP. 3' OH, that 3', what happens at 3' ends of DNA chains? They end, or you also can add on there. That is, in fact, this is where the next base is going to connect. Right? And if you look at DDC, what's at the 3' end? Is there an OH there? No. There's just a plain old H. Right? And if you look at AZT, is there an OH at the 3' end? No, there's this weird N thing, right? If you compare these guys. So the result of this is these guys have no 3' OH. Right? So you might think, oh, so what, right? Why is it missing it? Why does that matter? I want to draw you a picture for why that's really important. There are a variety of anti-HIV drugs that are missing their 3' hydroxyl groups. And the key, what makes that important is that 3' hydroxyl is where you connect the next base in the chain. So I want to show you why that would be bad news for a virus. So what I'll do is I'll draw it. Actually, maybe I'll go back. Write it down here. So these are called, so AZT and DDC are nucleoside analogs. And how do they work?